And I will start with thank you so much to Waters Wyatt for inviting me. I'm very happy to be here. I feel a bit like I'm the elephant in the room, a bit outside the box, but I kind of like it too. I'm French. So it suits me in one way or another. And it's quite funny also because we move from a very company, very specific question oriented to my talk will be more general about field for fractionation. I feel we have been talking about it, but not talking in detail. So I'm going to try to bring you into it. But I will start with very few words about SINTEF in Norway, because very often people are like, who you are? So fun fact, we are 2,400, so I'm not alone, but we are covering a bit like all the research fields you could think of, more or less. And we are a private, non-for-profit research organization working like half on publicly funded project from Norway or Europe or other countries, and 50% are more like industry-oriented project coming with specific question, a bit like CRO style, but perhaps a bit more like research-oriented. And yeah, we work a bit like at the combination of like academic, university, business, and us. And we were part of the very first EU project founded on LNPs. It was in 2010, I think, or something like that. And so over these past 14 years, we have been gaining experience. Many projects were publicly funded, so it was really nice to build competencies on it and to move towards like more company questions and like trying to find solutions. And that's what we are trying to cover these days. Moving to lipid nanoparticles, I think most of you know, but let's start from a quick background. The why you need an LNP is mostly because you need to stabilize the mRNA and to protect it from whatever degradation and to make it doing something when you inject it into the body, and then it will facilitate the cellular entry. One of the very specific things, it's like the RNA, if we stick on like mRNA or larger one, are extremely fragile molecules. So you need to protect them by putting them into an LNP, but then the LNP is also quite a fragile one, especially when it's with mRNA or longer ones. So you need to think about that. And one specific thing coming from the LNPs containing RNA is that the RNA is also a structural component. Without it, it will make the LNP being quite weak because you will be losing this like charge interaction between the RNA and lipids. Then when we move towards like, okay, you have a product, you wonder about like the drug product quality and the drug substance quality. But before going there, I like to say that nowadays it's seen like as a COVID vaccines, but behind, I think, given your company, you are quite well aware of it, there is a lot of things happening and a lot of questions, and many diseases are trying to be treated using such therapies. And many questions come with. If you dig into what USP and so on are providing us like the critical quality attribute, you can end up with such a list for the drug product. So more or less, let's say that there is 23, 25 different CQAs only for the drug product, mentioning 24 different techniques. It starts to be quite an extensive list to dig into to end up with something where you might know something at the end of the day. So the question we always ended up with when we talk to customers is like, okay, what do you want to know? Where do you want to go? And how? And also, what can we do for you? And it's always quite a question because all these things, such as particle concentration, size, polydispersity, RNA content from the LNP drug product, will all regard at the end of the day the safety, efficacy, potency, and stability. So then the question is like, which technique, how do you do that? And that's where I am bringing my best friend for close to 15 years now, the FFF aspects. So what it is, for you or for some which don't know, it's like chromatography-like. You just kick out the colon and you put a channel instead. And one of the main advantage is that you don't have stationary phase anymore. So you will have zero interaction between your particles and whatever you have in your colon. And given LNPs are quite fragile, you will be a bit more confident that you are not affecting, changing, or destroying your LNPs along the separation, which has been showed at the beginning of like the work with LNPs when we were trying the first sec colons, that the recoveries were really poor, and it wasn't like product related, it was separation related. So the multi-detector FFF aspect combines the separation in the channel. So you have an image here. Separation takes place in the liquid, and it's based on coefficient of diffusion. So we can see like size, the other way around of the sec, going from the smaller to the larger one. You can use a broad and wide range of mobile phases, which can suit your product and your analysis or your separation. But again, for LNPs, the best for the product. So you can stick with the buffers that the LNPs are containing in, which can be PBS or TRIS. Both of them could work because they are you can use them on FFF. And then it goes towards the separation, and from the separation to the detection. And here you can combine multiple detectors. A quick go through a separation. So here it shows the channel where you will separate your particles. And more or less what's going to happen is that you will have all your particles going at the bottom of it. And you will have a separation based on the diffusion. So let's say that the smaller one will go slightly higher in the channel and the larger one will go at the bottom. And then given you are in a channel, you will have a parabolic flow. So the smaller one will be eluted first, and the larger one will be eluted next. From that, you combine it with a cross flow, which is kind of sucking particles from the bottom to increase your separation power. Again, what you want to make sure is that your separation is great, but you are not affecting your product. So it's a game between different flows and conditions to end up with the best for the product and the analytics. We don't start from scratch, or we didn't start from scratch for LNPs. It's a technique used for a lot of years. It has been developed for nanodrug delivery systems, and especially it has been used for quite some years on liposomes. Pushed by FDA a couple of years ago, the liposome has been a big deal in the US, and at that time point I was working for the US government at NIST, and they were really asking, like, can you find out a method that would provide us a really good understanding of liposomes, especially the generic ones? Could we compare them with like the drug ones? So we work on it until we ended up with the first test method standards approved at ASTM, which is like kind of the little sister brother of ISO in one way or another, or the American version. So this method is now approved. So it shows that, okay, you can really end up with a solid standardized method for a drug product. So moving towards, what about RNA LNP? Can we do the same? And what can we do for that? Let's be the bad little duck for 1 minute. I think more or less everyone goes for the first thing. I want a size. I do batch DLS. But what you're gonna know about it with batch DLS? And I found these two examples in my lab. So that's DLS for mRNA LNP1, where you have a size of 70 nanometer and a PDA of 0.2 will be seen as good in the industry. I think it's fair enough. Then you have DLS number 2 on another LNP. It's quite the same. PDA is even better. And then you run an FFF on it, and you look at what's going on. So what you want to expect is like chromatography, one peak, one product, really good. So first product, I think it's really good, one peak, monodisperse, really nice. And then you put the second one, which had an amazing DLS at the beginning, and you end up with these very nice Alps, peaks, mountains, which I think show, okay, it is not as good as the DLS was claiming. So DLS is good, but what he says is questionable. So that's where I claim FFF can be more useful than batch DLS. So how can it be good? What can you do? And the advantage of is, okay, first the separation, but more than the separation, the next step is really like the detectors you will combine and what you're gonna get out of it. So very often people, or the best will be to combine light scattering, multi-angle and DLS to get molar mass, particle size, particle distribution, integrity, particle number, and morphology of your LNP. Using absorbance and array, you will dig into the concentration and composition in terms of RNA and lipids. All of that in like, let's say 1 or 2 hours. Quite amazing, but how do we do it? As for every single analytical techniques, you have to dig into a method optimization, which in that case, I would say belongs into these couple of different parameters. So the elution program, because in the case of chromatography, you will only play with like the flow, the main flow, while in that case, you can play with many different flows in the channel. And then the channel type, a bit like the colon type, you have the length, the size, and also the type of injection, kind of. the spacer, which determines the volume or the space in which the separation will take place, the membrane, it's a bit like the colon phase in one way or another, mobile phases, and then I placed here DCM because it's quite a question. It helps into hub concentration at the end of the channel. After digging into it, I decided to avoid bringing you into this perhaps not so fascinating method optimization. We got a quite optimized method, quickly summarized here, which has been a common work between our labs in CTEF and the Wyatt at that point. It was still only Wyatt, until the method was quite like robust and validated across a couple of LNPs. So then the next step was like, okay, can we apply it into like more like company or like product development questions? Then here you have a couple of examples on the batch-to-batch comparison. You can really see that some batches can be good, some batches can be different. It will tell you again about the polydispersity and the size of your product. On screening of formulation, I have an example just after, and the storage stability, which was and is still quite a big question in the LNP field. Which storage temperature, how long, how, and so on. And a quick example of like screening of formulation. So you can make plenty, you test them, and in like 1 hour, you will get quite a good understanding of like the polydispersity, quality, and different sizes, and so on. But the size is really good, but let's go a bit behind, and that's where here it has a lot of advantages. When you combine the light scattering detectors with UV and RI, you can dig into other things. And here you have an example where it's a loaded mRNA LNP and an empty one, where you can distinguish them because they will have like different size, but also coefficient of diffusion, so they won't be eluted at the same time point because their mass are also different. You will dig into the LNP morphology throughout the shape factor by using the multi-angle light scattering and dynamic light scattering. You will get the shape factor, which will tell you a bit about the shape. Is it like a sphere, loaded or empty? Is it like more rod style? Using the MALS, you can dig into the LNP number, particle concentration. In that case, we have compared it with the batch NTA, and they show that it is really close, within like, let's say, 5%. And finally, and I think it's perhaps the most interesting piece, it's really digging into the composition of it. By using this combination of detectors, you will dig into like, okay, how much RNA do I have in my sample, but also if I have different peaks, you can understand, okay, this one contains more, and the ratio between lipid and RNA will be that compared to this one. So it gives you a lot of understanding of your product. So let's say like an hour and a half, and you end up with 6 of the CQAs you are supposed to look into. As said just before, it's not GMP approved or like yet, but still when you are at the R&D phase and the product development and so on, it can be really useful and successful. But I would say there is quite some things behind that can make everything complicated, to my taste at least. It's quite a technique which requires skilled and experienced operator, given that it combines 4 to 6 techniques in one, from the FFF separation to the MALS, the DLS, the UV, the RI, and being able to do the data analysis in a very structured and qualitative way. If you're using size, people will complain that it's time consuming. Yes, an hour versus a minute can be, but you can get way more knowledge after all. It can be seen as expensive compared to DLS or other techniques. If you want RNA, you can go for RiboGreen. It can be way cheaper to do, but then here it's expensive, but it gives more. And no standard SOP yet on LNPs, and we are currently writing a ASTM standard test method for RNA LNPs using this method. So all in all, I like to say that it is a perfect tool for gene therapy and mRNA LNPs. And the main aspect to me is really that it doesn't affect your product itself. And that should be the thing that should drive people toward using FFF instead of other techniques which might affect your product. The method has been validated and across the various type of LNPs containing siRNA, mRNA, self-amplifying RNA. it's validated also across various labs and the 2 instrument platforms available on the market. And it's extremely relevant for academic research, but also to the industry. I had these slides yesterday evening because of Marlene points on like, okay, you are here, we can talk, so let's bring about like, what would you like to do to improve it? And here I am. I think the main thing here is really the data analysis can be or is seen as complicated. It's it's multiple steps, one after each other, which create multiple files, which makes things quite complex and messy in one way or another to end up with all the measurements I have explained. So you need to really know what you do in a very solid way. So it relies on a good knowledge of the FFF, the product, like the drug product you are working with, but the software. So you have a lot of manual software data treatments. You need to know how to do it. Whatever you do, at the end of the day, the software will give you something. No doubt. It's very rare that it doesn't give. So it's more to you as a user to know how to take this data and can you trust them. And one last point, I think okay, better transferability might not be the best wording of it, but currently there is 2 instrument platforms available. And compared to people working on LC or SEC, you can take a column from Weaver and put it on the HPLC from Weaver, and you can do something. In case of FFF, you can't do such a thing. Everything which is from Wyatt Waters can only stay there in terms of separation. Whatever which is from PostNova as the other brand has to stay there. And on top of it, the separation devices are not equivalent anymore. Things have been changed between the 2 companies, which makes things, when you want to standardize, extremely complex. What are you gonna say? Like, should I say, okay, the standard is only with Waters? Okay, PostNova will be pissed. Should I say, like, you have to do it only with PostNova? Oh, Waters will be pissed. Should I develop 2 methods? Yes, but my company is not willing to pay me forever to develop methods. So it makes things very complex at the end of the day, and that's something that company or people using FFF are complaining kind of because, oh, you talk to whoever user, I have this system, oh no, you have this one. How do we talk? How can I bring you the method I have in my lab into your lab? Stop complaining, conclusion. It's an amazing tool. Again, I think it really gives a way better understanding of the product in an hour or 2. Very powerful for the nanomedicine and gene therapy field. The combination of detectors is amazing, and I really hope in the coming months and years, even more detectors will come offering, again, better and better understanding, given you have a great separation before. And I will push for the fact that there is still a huge need of like method optimization, development, and standardization to hand up to a product that people product, like a recipe everyone can take and play with. And yes, I will finish with like, I consider it as a perfect tool for gene therapy. Thank you.